Partha was, of course, first of all, a friend, and also probably the mind I admired most was clear and crisp, deep and broad, and I could ask him almost anything in all the areas I know, and I know quite a bit in a shallow way in quite a number of different areas, and he could explain to me very clearly almost everything in all of them. And also, he knew very well what he did not know, which is sometimes a rare quality. Um, I would have loved to tell him about the work I'm going to describe today, because I would have loved to have his gentle and insightful and helpful criticism. This work is something I'm quite excited about. If this were biologically true, I think it would be quite an important advance in figuring out what cortex does, visual cortex does in particular. And I think it would be also the first time that a theory, a bit in the style of physics, would show to be working in neuroscience. The theory, as I will try to describe, seems to predict from one assumption about the computation of a part of visual cortex, seems to be able to predict the architecture, the function, and some of the aspects of development of cortex. So I think it's too nice to be true. So it's probably wrong, but let me tell you about it. Keep in mind that this is work in progress, and so it's not a mathematical theory. It's, several of the steps that I will mention, going from one result to another, are really motivated by some of the biology underneath, and I may not be able to explain all of this if you don't know it. There are also some gaps that need to be filled, although I think they are mostly details, apart from perhaps one. And again, if this were true, it would be scope for some interesting mathematics. Now, before I forget, I'll show it again at the end, but these are some of the people involved in work over the last 10 years that is behind the new work I'll tell you. Particular Steve and Lorenzo are here. And okay, the story is about this part of visual cortex, which is called the ventral stream. So this is the visual cortex of a macaque monkey. The ventral stream is this sequence of parts of cortex, of visual areas, named for strange reasons, V1, V2, V3, V4, IT. This is the back of the head. Its primary visual cortex is why Huber and Wiesel did their work that earned them a Nobel Prize 30 years ago or so. This part is the highest level for object recognition. This whole ventral stream is supposed to answer the question, what is in the image? This shows some of the connectivity between the different areas. What is interesting in this diagram, which is a recent one by De Simone and Movshon, is that it shows the size of this rectangles is proportional to the number of neurons in the area. Just to give you an idea, V1 has about, in the macaque monkey, 200 million neurons, same for V2. The total of the visual cortex is probably around 1 billion neurons or so. lot of connections, almost everything is connected to everything else. And the traditional, more or less, wisdom accumulated over the last 40 years by physiologists that put electrodes in the brain of monkeys and record what single neurons among these 1 billion neurons do, is roughly the following, is that in V1, you find neurons that essentially respond to a small part of the receptive fields, maybe a circle with a diameter of, say, 1 degree or so. My thumb at this distance is about between 1 and 2 degrees of visual angle. They are maximally stimulated by a bar of a certain orientation in a certain position. Looks like they're more or less doing something like a filtering with a Gabor filter, okay? Oriented Gabor filters. And in V1, there are 2 types of cells: simple cells, which are pretty strict in terms of needing a stimulus in exactly a certain position and orientation in order to be activated, and complex cells that also have the same selectivity, are also stimulated by oriented bars or contours, but are more invariant to the exact position of the stimulus and probably the size of the stimulus. So the picture that emerges from recordings like this and in higher areas is that you have simple feature like Gabor filters, oriented bars in V1, and some more invariance at the level of complex cells. In V2, you have somewhat more complex features and features of increasing complexity as you go higher up. For instance, V2 and V4, you may imagine that you may have the combination, like the logical end of 2 or more simple features like that, so that you get not only simple lines, but corners or crossings or so. Now, this is very cartoonish, don't take it too seriously, but gives you a feeling for the kind of optimal stimuli that are needed to stimulate cells as you go from the early vision to later stages. And at the same time, what physiologists find is that the receptive field of the cells, as you go higher up, so is the area of the visual field in which you could present a stimulus and the cell will respond, increases as you go from the back to the front, and the invariance to position and scale also increases. Okay, so over the past 10 years, we have developed, and so had other people. I think Amit will probably speak later about something similar also. Okay, where is he? Will you speak about also hierarchical model? Okay, so this is one of many models that try to capture what is going on in the ventral stream. And in this particular model, which is available for download, you have millions of cells simulating simple cells, essentially Gabor filters of different orientation, different position, and then complex cells, and then combination of features. At this level, it would be V2, and so on. And up to the last stage, where at the top there is a classifier, for instance, and the classifier receives a vector of features from the last layer of this hierarchical architecture. And this vector of features is more invariant, much more invariant, and possibly more selective than the original image. So you can think of this piece of architecture as a pre-processing stage for a supervised learning stage. And so the way this architecture is, can be thought of is that there is learning in the sense that we had very simple learning, unsupervised learning of tuning of cells at the S levels, the simple cells levels, in principle including also this one. In practice, this was mostly hardwired. But this is unsupervised, it's done once, it's like development of the visual system when a baby animal develops, and then it's fixed, and then it can do different recognition tasks depending on the supervised training of the classifier, for instance, at the top. Okay, this model was developed in over the last 10 years, as I said, taking into account a lot of the data from physiology and anatomy, and the goal was trying to summarize those data, try to capture some of the prediction that you could make about behavior of cells at different levels, in IT, in V4, and so on. And as I said, it's one of many. It's probably the most biologically faithful. As I mentioned before, it has two basic operations, one which is a tuning operation. You can think of it as a Gaussian tuning or a normalized dot product in which the input X is filtered as a dot product with some template, like a Gabor filter, represented by synaptic weight W, a vector W, to give selectivity. And then there is also, to go from the simple to the complex cells, a hardwired pooling operation. So like taking the max over simple cells that have the same orientation but different positions to give some invariance to position and scale. And this could be a max, could be an average or a nonlinear average. So two operations are a bit like or and and so you have an architecture that is a kind of disjunctions of conjunctions. Now, the goal of the model was, as I said, capture what we knew from the physiology and the anatomy. And in fact, it seems that the model did reproduce a few things we knew to be true, like tuning and invariance property at the level of IT, subunits in tuning V2, and predict a few other ones, the red ones, that were partly result of work we did with physiologists, like, for instance, this one with Jim DiCarlo, and some of the others, like this work with Lamp and Förster. But this was what the model was supposed to be doing. What was a bit more surprising was that the model did also well in terms of matching human behaviors in tasks like this, in which you have to say whether for each image there is or there is not an animal. So you have a fast presentation. So this is in order to have your visual cortex working in a feed-forward mode, not allowing you to have eye movements or so. The model is not made to do that. And let's see, let me repeat it briefly, but it's pretty easy for people to, even in very rapid presentations, to say, yes, there is an animal, or no, there is not. Okay, so the kind of surprising things, at least for me, was that when you look at how well people do about 80% correct on a task like this. This was not exactly how the task was run. There was a mask, were a bit more complicated, but the model also does pretty much the same level of correctness, and also with the same qualitative behavior of doing essentially best when the animals occupy most of the picture, and worse when there is a lot of background and context. And also the images which are easy for humans are easy for the model, and vice versa. This is not a proof at all that the model is a faithful model of visual cortex, but it says it should be taken somewhat seriously. And the other thing that surprised me even more is that when people in my group tried it, especially Thomas Serre, tried to use it on natural images and compare it to existing state-of-the-art computer vision systems, it did as well or better at the time in terms of detecting and classifying different objects in the scenes. This was surprising because the model was not developed to be good at object recognition. It was really developed to be faithful to the properties of the ventral stream in the monkey. And here is another demonstration in which a version of this model, which would be closer to the dorsal stream, but very similar, has provided the core of a system that we are developing for classify behavior of a mouse in a cage. And that's important because in our institute at MIT, the McGovern Institute, and nearby institutes like the Broad Institutes and so on, which are involved in genomics and genomics of mental diseases and so on. There are many hundreds, thousands of mutant mice that are used as models of schizophrenia, autism, depression, and so on. And in the meantime, getting the genomics, the sequence of the genes and so on, has become very easy, very cheap, is very quantitative. Ironically, the behavior is the part that is difficult to make quantitative. So this is an attempt. Typically, the way it's done is that you have a technician taking notes from a video of how a mouse behaves. Here we have a system that has been trained to classify 8 different types of microbehavior, so like rear walk, groom, freeze, and so on. And then from the sequence of these microbehaviors over minutes, hours, you can actually characterize pretty accurately which kind of mutant you have, for instance. And then, of course, the promise is that combining this large database of quantitative reproducible phenotype data together with the genotype data, you could discover interesting correlation. But again, for here, the point I wanted to make is simply that this system also derived from what we know about the visual cortex, does as well as humans. Okay, so so far so good. The problem has been that forgetting even the biology, it's not. I've been quite frustrated in not getting, even after essentially 10 years of working on this kind of relatively simple feed-forward model, to understand how they work. It's kind of ironic situation, maybe typical in the future for neuroscience. You have a model that you can simulate, but even if you have it, you cannot understand how it works. It's too complicated. So, a few years ago, we decided really that we need a theory, and we started to do some interesting steps with Steve and Lorenzo and others in the group. But it is only this summer that I thought that there was a somewhat different take from the previous one in terms that to me anyway, may clarify what this particular architecture is doing and why it may work. So this is unpublished, it's still preliminary. There is a paper in an online archive, a draft. But people are interested, I'll let them know. So the main motivation is asking what is the main difficulty in object recognition, biological object recognition. And it is whether, for instance, in a categorization task, like you are given one dog and one horse, so very, very small numbers of examples, and then you have to decide when you're presented other images, whether the new image is a dog or a horse. Okay. Now, so difficulty here means performance as a function of a number of examples, training examples. So it's a really simple complexity of this task. And the question is the difficulty is more because you can be given an image from any kind of viewpoint, distance, size, rotation of the animal, or illumination, or is mostly because of the variability within a class. There are different cats, different dogs, different horses. So this is just answering only one part of this, and not even answering, giving an insight on one part of this question. Suppose you have an oracle that normalizes the image so that they're all from the same viewpoint, same distance, same scale. So the only thing that remains is the variability within the classes, which is done here. Then the question is, will the learning problem, the generalization problem, be still difficult or quite easy? This is just an empirical experiment that Joel, one of the students, did, in which it seems that the answer is, after you have done this normalization, the problem is very easy. You can, with one example of a dog and one example of a horse, just taking L2 distance of pixels, you get 70% correct. And with 8 or 10 examples, and using, for instance, well, they're pretty similar, both measures. But so this will tend to say that the variability in the appearance due to viewpoint is the main cause of the difficulty of recognition. There are other arguments. It's like if you compute how many different images a single object could generate, and distinguishable images, making some assumption about resolution, size of the images, and ranges of scales, and so on. Turns out is in this order, it's, say 10 to the 10 or so. Whereas, how many different cats or dogs there are? Probably, I don't know, 1,000 that are different from our effectively distinguishable for us. So anyway, that's motivation. The basic assumption is that the goal of architectures like this is to provide a vector, let's call it a signature vector, kind of fingerprint for each image, for each object, which is as invariant as possible to transformations of objects. And of course, it has to be selective as well, enough to distinguish, but let's focus on the invariance part. So what I want to tell you is how the ventral stream could learn during development, how object transforms, how they change appearance because they translate, they move in depth, um, and so on, or you move, but during development, like first days, weeks, for a baby, human baby, and then how this stored images, because they're released or stored neural activity, can be used to get signatures that are invariant to those transformations, using mechanisms that are biologically very plausible. And I will deal mostly with affine transformations in 2D. And there are a lot of implications. I will go through them later for the theory. But one, for instance, is that features are not so important. What matters are really, from the point of view of what this ventral stream is doing, is which transformations is it invariant to. And one of the predictions, one of the many, but is that if you'd raise an animal or in an environment, artificial environment, people have done that in the past, but not in this way. The prediction is, from the theory that I'm going to explain, is that if the animal would be raised in a, say, random dots environment, just black and white dots, if the transformation to which is exposed, so like, moving around, would be normal ones, everything would be normal. But if you have a natural environment, like here, and you screw up the transformations, maybe by stroboscopic light, or in a virtual environment in which you don't have the normal translation, scaling, so then the development of the properties of neurons will be screwed up. Okay, so now let me try to explain, and I'll jump between two things. One is trying to explain how what happens during development, and for here I need motion sequences. And once this is done and it's frozen, then the problem I consider at the adult stage is the problem of seeing an object or an image, then being able to recognize, for instance, a face, and then being your face, and then being able to recognize your face in a second time, even if it's much larger or translated or rotate on the image plane or stretched or so. Good. Let me give you first an outline of how it's going to be. So the first one is the assumption that the computational goal of this part of the visual cortex is to learn transformation, really storing images of an object transforming, which means translating, scaling. Then I'll describe a little machine, a little process, which is what a simple cell could easily do, which is essentially to use the stored image as a template, dot product with a new image. You get a number, that's one component of this signature, and by doing this, you get a signature that is invariant to the transformations that the template were under. I'll be more precise. Then I'll argue that this will work, may require a storage which is too large, too many images to be stored as templates during development. And so there is a simple trick that evolution could have used, exploiting the fact that you can factorize the affine group as translation followed by a combination of rotation and scaling. And in this way, you can, evolution could have reduced from, say, 10 to the 10 to 10 to the 4 or 5, the number of templates that need to be stored. And this implies having a layered architecture. From this would follow the need of a hierarchical architecture in which you have receptive fields of increasing size. Because in the first layer corresponding to view 1, evolution would need to expose this first layer only to translations. How can this be done? I'll make the argument that if this receptive field, these apertures through which the visual world is looked at by the neurons, if the apertures are small enough, then each one of them will see basically translation only, independently of what's going on in the world. And then there is another assumption that there is plasticity of the synapses. So there is an unsupervised learning process going on, and which we know is true in cortex. And this, a quite general version of this has the property of computing essentially the principal components of the inputs in a non-line fashion. And so the consequence will be that the tuning of the cells, for instance, in view 1, will depend on the spectrum of these sequences of transformations, sequences of transformed templates stored during development. And the spectrum will depend not so much on the features or the statistics of the natural world, but on the transformations themselves. Which transformations? They will correspond to the characters of the group. And there are a couple of other things, if I have time. For instance, the fact that in this kind of reasoning, you need modules in cortex that are specifically dedicated to invariances for classes of objects like faces or bodies. I'll get to that. Okay, so let me start. First motivation is, it's just a motivation. So is the Johnson-Lindenstrauss theorem or lemma. It's just to make the point that in this architecture, we are not interested in reconstruction of the image, but in representing images in a way that they are distinguishable. And this results says that essentially random maps from, say, 1 million pixels space to a space of 100 features or so, 1,000 or less, is usually okay in terms of maintaining reciprocal distances between images. And furthermore, it says that random maps are okay. So in a sense, to me, this says features are not important as long as you have enough of them and you don't need too many. This is one just very concrete visualization of what may happen. You have an image represented here. This way, the image F with these components. D, as I said, could be 1 million. And you have here a number this is one template, a random template. I'm thinking, for instance, of one-dimensional images. And just dot products give you an image which has k components, and this tau templates could be random. Now, there is historical precedence for this. OCR 50 years ago, one of the techniques that were used were as simple as taking a random but fixed set of lines, superimposing on whatever you wanted to recognize, counting number of intersections for each line. This gives you a vector, a signature. Turns out if you have enough random lines, this works pretty well. And so, okay, so let me define this little machine that can give you invariance in a very simple, biologically simple way. Suppose that I have a set of images. These are images captured, for instance, a baby is looking around, something is moving. So it's a little movie, there is, and suppose I have a piece of the image, say the eye of the mother. This is tau 1, and tau 2 may be something else. Okay, now no movie. I could associate to a vector like this. This is my set of template. So when I want to use it, I can get essentially an instance of Johnson-Lindenstrauss in a sense. I can compute for a new image a signature in this way. So from a set of template, I just compute the dot products of the image F with each one of them. I get a number, so a vector of numbers. This would be the signature. And I do it for each of these templates. They're just patches of the images. But I can also, during development, each template, so tau 1, may be a patch of images that is moving. So I actually have a movie, a set of frames, okay, which are the product of, for instance, this object translated, for instance. And so I can have my this extended template book in which I have one image and the transformations under the action of G0, G1, which are elements of a set of transformation, possibly elements of a group. And by the way, transformation, what I mean is a number of things, but for geometric transformation, there should be parentheses here. If I have an image which is a function of x and y, the transformation of this by translation or rotation will be the image defined on the transformed coordinates, okay? And as I said, I will mainly consider this group, which is translation and then rotation and scaling. And so if I have a set of templates that represent all the transformations of a group, then there are a number of aggregation functions that give me a signature that is invariant to that transformation. So if I go back here, if I take the max over G from G0 to Gn, and then again the max here from G0 to Gn, I get for each one of those one number, and that number is invariant for here. Sorry, let me repeat. So here, I'll take the max, so take the highest number that comes out in these first 4 elements over the transformation, and do the same for the other templates. And so each one of these 4 groups will give me a number, and that number is invariant to the transformations that the templates have been subject to. So if I have templates that have been translated, and I do this operation, that this signature will be invariant to translations of the image F. Is that? Let me say that in a more formal way. Let's define images, dot products between images, for instance, F and the template G. These spaces in, say, L2, same for this template set, and then a space of transformations, and then if I consider these geometric transformations in which I transform the images by acting on the domain, if the Jacobian of the transformation is constant, then the following is true, is that this normalized dot product between an image and template is the same if both are transformed in the same way. And in particular, you have this result, which is the one I mentioned with the max, and not only the max, but other aggregation functions as well. Okay, so the part which is interesting here is that you need to have these templates that reflect the transformations, and they are simply images of an object undergoing a transformation. And then once you have that, you can have invariants for any new object that you don't need to have seen before. Okay, so this will work if you could store templates reflecting all transformations. As I said, this would be a fine in 2D, a 2 by 2 matrix operating on X, Y, and translation term. And but of course, you need to store a lot of templates because you'll need to take into account translation, rotation, scaling, and all combinations thereof. But you can factorize the affine group in this way using homogeneous coordinates now. So you'll have a part which is translation and a part which is rotation and scaling and asymmetric scaling. And so if you could use the same approach, I'm jumping a bit here logically, but if you can use the same approach of computing an invariant signature for translation, then you can later do rotation and scaling. And so if you do this invariance operation sequentially, then the number of templates that you need to store will be the sum of the two instead of the product. So you will save quite a bit of storage. But how could, so this says if you have a hierarchical architecture, at least 2 or more layers, and the first layer could take care of translations using this memory-based invariant module, then you could reduce very significantly the number of stored templates. But how could you expose the first layer to translation only, so that it could store only translation. Well, the one way to do it that evolution could have used is to use small aperture. Instead of looking at the full image, you look at a lot of small apertures, like small receptive fields, which is what happens in V1. And the claim here is that if you or a mechanism look at a small aperture, then almost anything, rotations or scaling, most of the time looks like translation. What was there, for instance, was a rotation, I think. And so, in fact, there are, just got yesterday a message from Andreas Maurer, who visited us, and he has a proof of this intuition that that depending on the size of the aperture, which is this ball V, you can approximate almost any transformation with linear terms. It's a really Taylor expansion. And so the argument is that evolution, by setting up this layered architecture with apertures of different size, can essentially select for learning during development which transformations are being learned. It can factorize the transformation. Certainly first translations and then the rest. So now there is another part, and there is a little disconnect here, which is a further way to reduce storage complexity, and which is the following: is let me see what comes. Let me explain it. So suppose you have now one of these apertures in V1, and you collect, during development, there are a lot of motions of different patterns, and so according to this rule I described, you collect all these movies, and by doing this max operation later, you have invariance to translation. But you could also, instead of just collecting all of them and keeping them, you could perform a principal component analysis on those, maybe to save, for robustness, to save storage. And interesting enough, we know that the synapses of those cells, this would be cells that become simple cells, the first cells in cortex, there is Hebbian plasticity. And Hebbian plasticity, I'll show it later, small variations of it is what is known as Oya algorithm or Oya flow, and it's basically an online learning techniques that computes PCAs online. So you have the machinery there to compute PCAs. And if you do this, when you move anything, random dots, natural images, and you look at the principal components through a small Gaussian aperture, you get essentially Gabor filters. So, and the interesting thing is what you get does not essentially depend much on the patterns, the structure of the patterns are moved, but mostly on the direction of motion in this case. So the transformation is what determines this eigenfunctions, and these are other examples. These are simulations, by the way. These are the kind of top eigenfunction you get, which is it's a top complex eigenfunction. It's a complex exponential. And so you get the real and the imaginary part, the cosinus and the sinus component of a Gabor filter. So it's a complex exponential modulated by a Gaussian. Okay, so, and for small apertures, even rotation gives you, because of what I said, that for small apertures, everything looks like translations. To a first approximation, you get similar Gabor filters. At higher levels, and this is one of the gaps, the gap I was mentioning, we still don't know exactly what is what the simulation will give as we go to the next layer, but with larger receptive fields, you will expect that they capture something else than translations. For instance, scaling would be my guess. Scaling will have invariants that be more like this. Essentially, these are the orthogonal to expansion. It looks like we'll be getting receptive fields that are somewhat similar to the ones that physiologists have measured at in higher areas of the ventral stream. But this is work to be done, or is going on right now. And this is, by the way, the motivation for, the Hoya rule, in which the weight of a synapse changes. Is essentially a gradient system, is online stochastic gradient descent, in which the weight of a synapse, this was the original Hebb rule, changes depending on pre and postsynaptic. This is the postsynaptic firing, this is the presynaptic input, and in the Hoya rule there is an extra term here that you need to normalize this process to avoid that it blows up, and there is evidence of not only of this part, but also of the normalization part in visual cortex. So this is related to the characters of the subgroups that make up the affine group of transformations. So you have translation, rotation, symmetric dilation, and each one is a locally compact abelian group, and although it may not be abelian on the whole, but locally it is, and then essentially to each one by themselves, each one of these groups, you have characters of a Fourier description that is in terms of complex exponentials. And there are some interesting questions when you have a transformation that is composed of two transformations that don't commute, like translation and rotation, because then you have we don't have common eigenvectors, and there is some interesting question of what happens when Hoya rule will to what will converge exactly. Okay, let me converge myself. I'll just mention that the machinery that we have used, that I mentioned at the beginning, for invariance, can work approximatively, not exactly, also for not affine transformations. So suppose, for instance, that I would like to, give you a face once, frontal, and then I want you to try to recognize an image of the same face rotated in depth. This is not an affine transformation. And in general, for an object, it's impossible to do something like that unless you know the 3D structure, or you have more than one image. But suppose this case, if I use the same machinery in which I collect templates from another face, rotating in depth, and I do what I said about dot products and taking the max, then I'll get much more invariance than otherwise. So this is how well my recognition works as a function of the second image being rotated relative to the first one in depth, just doing nothing special, the normal model. And here I get much better performance, not 100%, but much better by having this invariance module. Okay, so let me stop here. Here are some of the various predictions. I mentioned already some of them before. Thank you.
Features where you get the products are taking the max. So while it's clear if the invariant features do not are complete.
Sorry?
Do not are also complete in the sense that if you have two images that are not translation of one another, you have at least one feature which is different.
Well, this depends, of course, on the features. I can choose, stupid features or very few of them, and they will not be discriminative. I'm assuming that I'm starting with enough random features to have, something like Johnson-Lindenstrauss working, right?
Yeah, but is there a proof these are complete?
I'm not sure I need a proof, right? Anyway, this is, I just take what, our experience says more or less random features work. We did, like other people did in the past, we learned features from natural images. They work, thousands of features. That then it started to appear that if we take random features, they work as well, which is more or less what we expect from. And so provided you have enough of them, they're not stupid.
You're not taking random features, you're taking random images and taking your SVD. That's not random features. You're learning very stable structures among the random images.
No, we have used the completely random images with no difference. Well, I'm not saying, let me see, I'm not saying that features cannot be, one could improve performance by choosing, carefully features. I'm just saying it's not a big deal. This results, this is natural images. I have another slide in which we have random dots and same results. Let me see if I actually have it here. Yeah.
The local statistics of invariant fields, if it's random dots or smoother things, give the same high eigenvalue components, but you're learning these things.
Well, but this is reflecting mostly the transformation, not the pattern. That's all I'm saying.
Many animals are born extremely precocious in the sense that they, having had presumably no vision at all, they come out of the womb and immediately get up and walk and act like they recognize all kinds of objects and things. What's your comment on that?
Yeah, we still don't know, how much comes from the genes and how much needs visual experience. So far, in primates, it seems it's a combination of both. So that, for instance, if you raise a cat in an artificial environment, development like stroboscopic light, development of the properties of the cells is affected quite strongly. But for instance, there is a somewhat crazy alternative to the translation story that I told you, which is the following. There seems to be retinal waves during development, before birth, which are essentially waves of chemicals, but giving rise to electrical activity in the retina, say of cats. And these waves may be quite planar in a sense. And, anyway, probably does not matter too much, but this may give rise to be equivalent to translation at the level of V1 during development, so that you actually don't need exposure to actual visual world. There are many things like that are between genes. Basically, it's genes in this case, not the environment, but kind of simulating what we may need in the environment for development. And yeah, there are many animals. I mean, the general rule seems to be the simpler the animal, the mostly already hardwired it is by the genes themselves.